Bismillahirrahmanirrahim, and welcome back to this next video, and this is the 12th video of the chapter 2 of Immunology. From the last couple of videos we are talking about the granules of the neutrophils. First we talk about the primary granules and its composition. The primary granules are also known as the Azurophilic granules, and then we are discussing the secondary granules, which is also known as the specific granules in the neutrophils, and we have discussed a lot of the components that are present in the secondary granules. Now this particular video is the continuation on the specific granules or the secondary granules, and in this particular video we are going to focus on two other antimicrobial components that are present in the neutrophils. The first one that we are going to discuss is the elastase. So I'm not going into the detail on the functions of the elastase, because this elastase that is also present in the primary granules, and I have a detailed video on that when we were discussing the primary granules or the Azurophilic granules, and I'll share the link in the description. But as these elastases, they are also present in the secondary granules, so I'm going to give you an overview how this elastase its work. Now this elastase, they are enzymes. They belong to the serine protease family, and what they usually do is that they are going to target the amino acids that are present in the cell wall of the bacteria, and they usually target the alanine amino acid. Now what this elastase do, that it is going to break down the peptidoglycan layer of the bacteria through its proteolytic activity. And if you can see the structure of the cell wall of the bacteria, they have got these NAG and the NAMs. They are connected to each other through the beta-1,4 linkages, but these chains, this one chain of the NAG and the NAM, this other chain of the NAG and the NAM, they are also connected to each other by these short peptide sequences, and these short peptide sequences are providing the structural stability to the cell wall. What the elastase do, they are going to break down these short peptide chains, and when they break down these short peptide chains, that is going to make the bacterial cells osmotically unstable, and when they become osmotically unstable, the cell eventually bursts due to the influx of the water. That means that the elastases, they've got a very strong antibacterial activity, and when the osmotically unstable bacterial cells, they are there, they actually lead to the death of the bacteria. In this particular video, I mostly want to focus on the cationic antimicrobial proteins. For short, you can call them as the CAPs. And they are very important components of the secondary granules of the neutrophils. When you talk about these CAPs, CAPs that include the defensins, the lysozymes, and the important cathelicidins. We have already discussed on the defensins, the lysozymes, but I want to focus on the cathelicidins. The videos on the defensins and the lysozymes, I'll also share the link in the description. But in this particular video, we are going to focus on these cathelicidins. When you talk about these cationic antimicrobial proteins, why they are called is the cationic antimicrobial proteins? Because if you look at the overall charge on these peptides, they actually carry a net positive charge. Therefore, we call them as the cationic antimicrobial proteins. Now, these cathelicidins, they are a family of the antimicrobial peptides, and they are stored in the neutrophil granules as inactive precursors called procathelicidins. That means they are produced in their inactive form, and when they are needed, they are then converted into the active form. This would be very much similar to the enzymes or the proteases that you produce in your digestive system, like the trypsin, they are produced in the form of the trypsinogen first. When they are needed, they are then converted into the trypsin, the pepsinogen, for example. That would be another example. There the pepsin that is produced in their inactive form, known as the pepsinogen, and when they are needed, then they are converted into the pepsin. Same is the case over here, that these cathelicidins, they are produced in their inactive form, which are known as the procatelicidins, and when they are needed, then they are going to get activated. Now, when they are needed, they are actually activated by the proteolytic cleavage, and this proteolytic cleavage, what it actually do is that it is going to remove the propeptide region from these procatelicidins, and when this propeptide region that is removed, that is actually removed via proteases. That depends on the type of the cells. Sometimes you can also use the serine proteases. So when these procatelicidins, they are activated by proteolytic cleavage, by the removal of the propeptide regions, these cathelicidins, they are actually converted into the active peptides, and these active peptides can do two things. They exhibit the antimicrobial activity by disrupting the microbial membranes. We will see that in detail how this is happening. And these active peptides, they are also responsible for modulating the immune response. For example, they can help in the release of the cytokines. Now, these cathelicidins, they exhibit broad spectrum antimicrobial activity. By that I mean that they are not only active against the bacteria, but they are also active against fungi and some enveloped viruses. When we talk about the human beings, the most extensively studied cathelicidin, that is known as the LL-37, and if you look at the net positive charge on this LL-37, that is usually 6, therefore we call them as the cationic antimicrobial peptides. This LL-37, that is actually derived from the human precursor protein, which is known as the HCAP18. This H is for the human, this cap, as I've told you, this is for the cationic antimicrobial protein, and this is 18. So this LL-37, that is an important example in the human beings when you talk about the family of the cathelicidins. Now, how this LL-37 it works? I'm focusing on the functions of the LL-37, but it applies to all of the cathelicidins because they have more or less the same mode of action. Now, the cathelicidins, like the LL-37, they possess an amphiphatic structure. So their structure, they contain the hydrophobic as well as the hydrophilic regions, and when a structure contains both the hydrophobic and the hydrophilic regions, we call them as the amphiphatic structure. So the LL-37, because of its amphiphatic nature, it allows them to interact with the lipid bilayers of the microbial membranes. Now, what happens is that upon encountering a microbial membrane, this LL-37 that is going to insert itself into the lipid bilayer with its hydrophobic region buried within the lipid acyl chain. This is hydrophobic, this is hydrophobic, like dissolve like, that applies. So the hydrophobic region of the LL-37 that is going to interact with the lipid acyl chains of the microbial membrane, and their cationic regions, that would be the hydrophilic regions, they are oriented towards the membrane surface. Now, the interaction of the LL-37 with the microbial membrane, that leads to the formation of pores, and when the pores they are made in the microbial membranes, that actually compromises the integrity and permeability of the membrane. And you all know that when the permeability and when the integrity of the microbial membranes that is compromised, that actually is responsible for the leakage of the intracellular contents. And when the intracellular contents that are released, that actually leads to the death of the microbial cell. Now, when we talk about these pores, this pore forming activity of the cathelicidins, like the LL-37, that is often associated with their ability to oligomerize or aggregate on the membrane surface, and by oligomerizing or by aggregating, they are making pores, and these pores are actually responsible for the leakage of the intracellular content of the microbial cell, leading to the formation of the you can say leading to the leakage of the content of the microbial cell and their death. Now, this LL-37, beside interacting with the microbial membrane, that can also interact with other targets in the bacteria, like the DNA, like the proteins, disrupting essential cellular processes. For example, if they are interacting with the proteins, that are responsible for the formation of the cell wall. So if they are interrupted, that would simply mean you will not be able to go for the formation of the cell wall. Now the question is that if the bacteria, they have got these membranes, the microorganisms, they have got the membrane, but at the same time the humans, they've also got the membranes. So how the cathelicidins that are produced by the humans, of course by the neutrophils, how they are going to differentiate between the microbial membrane and the membrane of the host cells. Now these cathelicidins, they exhibit selective targeting of the microbial membranes or the mammalian cell membranes. And how that is happening? Now this selectivity is actually attributed to the differences in membrane composition between the microbial and the mammalian cells, such as differences in the lipid composition and the surface charge. If you look at the microbial membranes, they typically contain higher proportion of negatively charged lipids, such as the phosphatidylglycerols and the cardiolipin. As I've told you, we are talking about the cathelicidins. They are cationic antimicrobial proteins. They have got an OR positive charge. If the microbial membrane, they have got highly negatively charged lipids as compared to the microbial membrane. So these cathelicidins, because of their positive charge, the microbial membrane, because of their negative charge, they are going to interact with each other. So this negative charge on the microbial membrane, the positive charge on the cathelicidins, that actually provide an electrostatic interactions. And when these electrostatic interactions, they are established, they actually facilitate the binding of the cathelicidins to the microbial membrane in comparison to the mammalian cell membrane, like the human cell membrane. So this is one of the reasons that we say that the cathelicidins, they are selectively targeting the microbial membrane. They are not targeting the mammalian cell membrane like the human cell membrane. Now this is the whole story that I've told you, that the neutrophils, they are going to produce these cathelicidins. These cathelicidins, they are going to act on the bacteria. If this is the membrane of the bacteria, as you can see over here, when the cathelicidins, they interact with them, that is going to make these pores. And when there are formation of the pores in the membrane of the bacteria, for example, that is going to lead to the leakage of the intracellular content. And this leakage is actually responsible for the death of the bacteria. So if you like the video, please subscribe to my channel, hit the notification bell. If there is any question, you can ask me in the comment section, and do share it with your friends. We will continue the discussion in the next video.